Chapter 3, Section 6, Glycolysis and the Citric Acid Cycle. And the glyoxylate cycle. Wow, I'm going to have trouble saying that one today. Glycolysis. Okay, you'll see this sometimes in textbooks and papers and whatnot, referred to as the Embden-Meyerhof-Parnas pathway. I do believe these are three individuals who are responsible for the finding and identifying quite a few of the steps and intermediates involved. This glycolysis, as you've learned since probably junior high or high school biology, the taking of glucose and catabolically breaking it down into pyruvate, a 6-carbon sugar breaking down into 2 3-carbon sugars. Excuse me. This is nearly universal, okay? This is something we all do, and I use the royal we there as in all living cells, pretty much, on this planet. I want to say that with a little asterisk after that, because are there cells out there that don't use it? Yes. Or what they use is a variation of, because they may not use glucose. Oh, yeah. Remember those lithographs? Do not use carbon as their energy source. They have to have carbon to build up, the amino acids, nucleic acids, all that stuff, because everything living on this planet is an organic organism, but not everybody uses organic molecules like we do as their energy source. Glycolysis, yes, you've learned since you were a kid. Glycolysis immediately into the citric acid cycle. But as we're going to be talking about here, no, glycolysis can feed into fermentation, aerobic or anaerobic respiration. Okay, it's not every organism goes by what you've learned in previous courses, like intro cell and molecular cell biology. There are always variations. So let's talk this glycolysis first. Okay, glycolysis, got to remember, 2 stages, phases, however you want to think about them. What our textbook here refers to as the preparatory. Other textbooks, other sources will refer to that as the investment phase, where you're going to take a sugar, a 6-carbon sugar, and you're going to start rearranging it, getting it ready to be split in half. Then you're going to have the second stage, where you get everything out. You're going to have the redox reactions. Remember redox, hot potato, who's got the excited electron, where's it coming from, where's it going, and you're going to end with 2 pyruvates, 2 3-carbon sugars. So when you look at it, and this is just everything in a nutshell, real quick, you can see down here, here's the intermediates, labeled A through I, and then 1 through 10 are the enzymes involved in this. And you can literally just split it in half. Stage 1, above the line. Stage 2, below the line. Stage 1, you take a 6-carbon sugar that has 6 sides, and here's 1, 2, 3, 4, 5, 6. Carbon 6 sitting up there like a flag above the plane of the sugar. Add in energy, the third or the gamma phosphate from ATP. Okay, why do we need to add in the energy? Because step 2 here, we're taking our 6-sided ring and we're turning it into 5 carbons. Still 6 carbons total. It's now just 5-sided. 1, 2, 3, 4, 5, 6. And then we're going to add in some more energy. Why? Because we're getting ready to do a split. Going to split this in half. So here's energy to make the new ring shape. Here's energy to split everything in half. Now you have 2 halves. Problem is, you look at D and E. D, dihydroxyacetone phosphate. E, glyceraldehyde 3-phosphate. Only G3P is usable. Dihydroxyacetone phosphate is an isoform, Has the same number of carbons, hydrogens, oxygens, phosphates, all of that. It's just they're not the same, okay? They're isomers of each other. Enzyme 6, right here. Glyceraldehyde 3-phosphate dehydrogenase. Its job is to take G3P, also known as glyceraldehyde 3-phosphate, add a phosphate to it, strip off some excited electrons. G3P dehydrogenase does not recognize dihydroxyacetone phosphate. So these two right here, G3P, dihydroxyacetone phosphate, they go back and forth. Triose phosphate isomerase is an enzyme that takes and just changes the shape back and forth, back and forth. Whenever the shape becomes G3P, this enzyme takes it, adds a phosphate, moves it along. Dihydroxyacetone turns into G3P, moves along. And then you have the second stage where you get the energy out. You add on, now notice it does not say ATP here, it just says phosphate. So you have some free-floating inorganic phosphates. When I say inorganic phosphates, that means PO4, okay? PO4 added on, and then immediately taken off. Boom. Added on, taken off. Added to ATP. Now, since it's an enzyme, stripping a phosphate off and then adding it to a diphosphate, A2-phosphate, they refer to this as substrate level phosphorylation. It's enzyme driven, so it works and it works really good. But the thing is, it's not quite as fast, it's not quite as efficient as what you've learned about in cell and intracellular with oxidative phosphorylation in the mitochondria with ATP synthase. But still, it's 2 ATP. Why 2? Because remember, this gets split in half. You get a dihydroxyacetone phosphate, G3P. G3P goes through. Dihydroxyacetone gets turned into G3P. That one goes through. So for every one glucose, you will ultimately get 2 G3Ps. Well, after this, it gets turned into G3P. They both go through. So that's why you see 2, 2, 2, 2. Both halves of the original glucose will then be sent through this process. Substrate level phosphorylation. And then notice, basically everybody stays the same for the next 2 enzymatic reactions. It's just their positions move around. Rearranging the furniture. Move, move. And then the final step. This right here, I, is known as phosphoenol pyruvate. PEP for short. Phosphoenol pyruvate, it's hard to say, so we give it the acronym PEP. PEP will have its phosphate stripped off, added to a diphosphate, substrate level phosphorylation, and you end with 2 pyruvates for every 1 glucose. 2 ATPs go into this process, 2 NADHs come out. There's excited electrons right there. Remember, in biology, anytime we say electrons, we mean hydrogen, because remember, hydrogen's nothing more than an excited electron carrying along a proton. And then 4 ATPs come out. Cost you 2, but you get 4 back for a net of 2. And basically, here's everything I just talked about. Glucose, G6B, glucose 6-phosphate. Glucose 6-phosphate gets isomerized to fructose 6-phosphate. Why do we say glucose 6-phosphate and fructose 6-phosphate are isomers of each other? Count up the carbons, count up the hydrogens, the oxygens, the inorganic phosphate. It's the same number. They have the same molecular formula. It's their structures are different. 6 carbons forming a 6-carbon or 6-sided ring, 6 carbons forming a 5-sided ring. Add in another phosphate, fructose 1,6. Remember, carbon 1, carbon 6 now have phosphate groups. The addition there, powers this aldolase, splits it in half. Glyceraldehyde 3-phosphate, which you keep hearing me refer to as G3P, and then dihydroxyacetone phosphate. Dihydroxyacetone phosphate gets turned into G3P, and then G3P gets carried on. Reduce, NAD. Remember when you add the electron to something, it gets reduced. G3P gets oxidized. To be oxidized means you lose the electron. Here's who gave it up. Here's who got it. And then you synthesize ATP, substrate level phosphorylation. Go through, move everybody around, rearrange until you get PEP, phosphoenol pyruvate, which ultimately becomes pyruvate. So, use 2 ATPs to power everything in stage 1. And then the second half, you get 4 ATPs back and 2 NADHs with 2 pyruvates that can now be used elsewhere. One of the places primarily it's going to be used is the citric acid cycle, commonly referred to as the CAC. You go back into older textbook, you'll also see this referred to as the Krebs cycle. Krebs, the guy who figured out most of it, so they gave it his name. There's now this push now to get rid of people's names on stuff. Too egalitarian, I guess. I don't know. I figure if you figure something out, you should be able to name it after yourself. Anyways, now they're referring it to as the citric acid cycle. The citric acid cycle is going to be a means for consuming pyruvate. Now here, when biology, and we're talking respiration, when we say something is consumed, we don't mean eaten, we mean broken down, cut up until its individual components. Here, pyruvate will be consumed so that the 3 carbons making pyruvate will be released as 3 molecules of carbon dioxide, CO2. For this to occur, for pyruvate to enter into, the carbons of pyruvate to enter into the citric acid cycle, it's going to have to be attached to and formed what is called acetyl-CoA. Acetyl-CoA is a 2-carbon strand. So that means the first carbon dioxide of pyruvate has to be released. An NAD gets reduced to NADH. Acetyl-CoA will enter in and bind to oxaloacetate. 2 carbons of acetyl-CoA, 4 carbons of oxaloacetate becomes the 6-carbon citric acid cycle, which is what the cycle is named after. Going through round and round. And I say cycle, remember in science, whenever we say something is a biological cycle, that means there is no beginning, there is no ending. It's this constant churning of enzymes and substrates and products and intermediate forms, and over and over and over. Acetyl-CoA gets fed into the system, this cycle. And as those carbons, those 2 carbons for acetyl-CoA are involved in and swirling around in this, take it a couple of runs through the cycle, and then those 2 carbons will be released as carbon dioxide. 3 NADs will be reduced to NADH. 1 FAD will be reduced to FADH2. And 1 ATP will be formed. And what's interesting about these cycles, you add in the acetyl-CoA, you form citrate, and you go through, boom, boom, reduced, reduced. Here's a carbon released. There was one released. Here's another one released. And it just goes through the cycle over and over, releasing, releasing. ATP formed. And then you get to this part here where, you know what? We need to remake oxaloacetate. So you take succinyl-CoA, strip off the phosphate to make succinate, and then you will oxidize it, reform the carbon strand, move around the bonds until you remake oxaloacetate, which can then bind to another acetyl-CoA, and it starts it all over again, and again, and again, and again. When you look at the structural formula of alpha-ketoglutarate and oxaloacetate, you look at these and you squint real hard, you'll see that alpha-ketoglutarate and oxaloacetate kind of sort of resemble some amino acids, and there's a reason for that. They are what we call the precursor. They are the building block. Oxaloacetate, alpha-ketoglutarate will be pulled out of this mix of the citric acid cycle, and then will be used to make and convert to make amino acids. Some of the amino acids in the proteins of your diet will be broken down so that they are now alpha-ketoglutarate and oxaloacetate and then fed into the citric acid cycle so they can be consumed. And, the excited electrons used to reduce NAD and FAD. Succinyl-CoA is a precursor for the making of the cytochromes and chlorophyll. Acetate, which is, remember, the acetyl-CoA, acetate being the 2-carbon strand, can be used for making fatty acids. How can it be used to make fatty acids? 2, 2, 2, 2, 2. Next thing you know, you've got 16 to 24 carbons forming a hydrocarbon, and ding, 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 that's a fatty acid. Attach at least 2 of them to a glycerol and throw on a phosphate group, you got a phospholipid. Throw on 3 of those fatty acids to a glycerol, now I have a triglyceride. There you go. Nothing in these metabolic pathways that we're talking about in this chapter occur in a vacuum, meaning they occur on their own with nothing feeding into them, nothing coming out of them. I mean, just here on this slide here, we see that amino acid synthesis, fatty acid synthesis, cytochrome, the things that are going to be used later in oxidative phosphorylation. Those can take parts of the citric acid cycle out and use those as building blocks in anabolic metabolism. Just as the things you eat in your diet, the proteins, get broken down to polypeptides broken down into amino acid, and some of those amino acids feed into the citric acid cycle. The phospholipids, the lipid portion, the fatty acid portion, cut, 2 by 2 by 2 by 2. Throw on a CoA to each of those. They look exactly like acetyl-CoA. They are acetyl-CoA. Feeds directly into the citric acid cycle. So, glucose, citrate, malate, fumarate, succinate, all of these can feed into glycolysis. It's just at what point can they feed into glycolysis? Can they feed into the citric acid cycle? Glucose, we always refer to glucose when we're talking about these 2 pathways because glucose starts at step 1. Any of the other sugars or carbon strands that are C4 to C6, 4 carbons at 6 carbons in length, can feed into. Some organisms can utilize acetate as a direct carbon source. They're going to utilize acetate in what is referred to as the glyoxylate cycle. Okay? It's going to use some of the same enzymes in the citric acid cycle, but it's going to also utilize new enzymes referred to as isocitrate lyase and malate synthase. What this does is it takes C2, and instead of linking them together to make a fatty acid, it's going to link them together to make a carbon chain that's going to resemble one of those intermediate steps we've talked about a few slides ago. So acetate can become acetyl-CoA if it's there. There's enough of them. which can then be added to citrate, be added to oxaloacetate, sorry, to make citrate, fed into the citric acid cycle, so it can be consumed that way. Sometimes, though, the isocitrate needs to be fed back, reversed, becomes glyoxylate. Glyoxylate, along with an acetyl-CoA, becomes malate. Malate becomes oxaloacetate, which can then bind, make citrate. Isocitrate gets reversed, becomes succinate, and can then be used in many other things. A 4-carbon strand, a 2-carbon strand come together to form a 6. Glyoxylate with acetyl-CoA, 2 and a 2, make malate. Malate gets reformed to make oxaloacetate. Oxaloacetate, which will bind with other acetyl-CoA to form citrate. So it all depends on the organism and what carbon source it has available. Not all organisms can do this. Okay, to be able to do this means you have to have the genes for the enzymes that control this.